friends, this is Dr. Paushali Pal here explaining to you antibiotic resistance of bacteria. Now before I go into the topic, let me tell you why did I take this topic. This is very much pertinent in these days. As we have got so many microorganisms in the air these days, especially in the days of COVID-19, you may want to create a novel drug, an antibiotic against the bacteria. For that reason, you need to have the knowledge, basic knowledge about antibiotic resistance of bacteria. So, the bacteria, what happens? What is this antibiotic resistance that I am talking about? When a bacteria is killed by the antibiotic, it is said to be sensitive to the antibiotic. Now, what happens when the bacteria is not killed? It is said to be antibiotic resistance, because the bacteria is able to prevent itself from the drug. Antibiotic resistant it becomes. Okay. Now, there are several mechanisms by which the bacteria can prevent itself from the different kinds of antibiotics. Okay. To some extent, the bacteria are naturally resistant to all the antibiotics, because a single bacteria cannot be killed by all of the antibiotics, or all the antibiotics cannot kill all the bacteria. So, bacteria to some extent are especially resistant to the antibiotics. But when it comes to a greater gene pool, we consider this concept as antibiotic resistant. Okay, so now let us go through the several mechanisms by which the bacteria prevent itself from the antibiotics. I'll tell mnemonics for this. I have used a mnemonics, that is LIDAR. This is my mnemonics. You can remember this in times of exams also. That is leader. Don't go by the English spelling. It is LIDAR. L stands for lack of structures. Bacteria adopts a mechanism. Bacteria lacks structure. Bacteria lacks structures. It's a mechanism of the bacteria. Okay, what happens here? The mycoplasma is one genera of bacteria in which the cell wall is not present. So, the antibiotic penicillin, which acts on the cell wall of the bacteria. Suppose the cell wall is present around the bacteria like this. The cell wall is not present means where will the penicillin act? The cell wall is not present. So, where will the penicillin act? The penicillin will not be able to act in that case. So, the mycoplasma becomes resistant to the penicillin. Got me? Another mechanism, this is L, lack of structures. Next is I. Bacteria is impermeable. To antibiotics. That is, the bacteria, within the bacteria, the antibiotics are not able to enter. Say some gram-negative bacteria. This is a gram-negative bacteria. Suppose this is a gram-negative bacteria. Within this, the penicillin G tries to enter. If it tries to enter, we provide a drug penicillin G. If we provide the drug to gram-negative bacteria like E. coli and other enteric bacteria, the bacteria does not let this penicillin G enter into its cell. That is, the bacteria is impermeable to the antibiotics. It is no entry for the drug. So this is a way in which the gram-negative bacteria saves itself from penicillin G. This is another mechanism. We have finished L, we have finished I. Now next is D. D stands for deactivation of antibiotics. Bacteria deactivates the antibiotics. What happens here actually? Some bacteria like Staphylococcus, what happens? They have the enzyme beta-lactamase. I'll draw the structure of penicillin to explain. This portion is the beta-lactam, and the beta-lactamase, that enzyme, acts on here like a scissor. It acts like a scissor, as I have shown you. Okay, it cleaves the beta-lactam ring, and therefore this whole structure is of penicillin gets deactivated. This whole structure, that is penicillin structure, gets deactivated because of this enzyme beta-lactamase, which is present in Staphylococcus. Okay, so this particular enzyme is responsible for cleaving the drug penicillin and thereby saves the Staphylococcus from the drug penicillin by deactivating the drug. Next is, we have finished with L, I, D. Next is A. Bacteria alters the target. Alters the target. What do I mean by this? Target means the place, the site at which the bacteria the antibiotic acts on the bacteria. Suppose the target is peptidoglycan of the cell wall. It's having the sugar moieties as NAM NAG, that is N-acetyl muramic acid and N-acetyl glutamic acid. Pentapeptide is attached like L-alanine, D-glutamine, D-aminopimelic acid for E. coli. This is the E. coli pentapeptide structure that I am drawing, peptidoglycan, alanine, D-alanine, again D-alanine. Now as you can see here, this D-alanine is the terminal peptide. This terminal peptide is converted by the bacteria to D-lysine or D-serine. So a drug called vancomycin can recognize this vancomycin drug when administered to this E. coli, it cannot recognize this D-alanine because this D-alanine has been converted to D-lysine. So that saves the bacteria from lysis. Otherwise, if the vancomycin would have recognized this D-alanine and have lysed this peptidoglycan, that is the cell wall of the bacteria. But in this case, the cell wall of the bacteria is saved only because the vancomycin is not able to recognize this D-alanine because it's not in place. Instead, D-lysine is in place. So the bacteria saves itself from vancomycin. This is another way bacteria alters the target. Now we have covered L, I, D, deactivation, A, alter the target. Now left with resistant pathways. This is the fifth point. Resistant pathways are for resistant pathways. Here what happens in resistant pathways, the bacteria, suppose you can say the sulfur resistant bacteria, sulfur drug resistant bacteria like staphylococci, what it does, folic acid is synthesized by, folic acid synthesis is blocked by sulfur drugs. So the bacteria is very smart. What it does, the bacteria actually acquires the folic acid needed for its nucleic acid synthesis from outside the environment. Okay, and therefore the folic acid synthesis is not required to be done by the cell, bacterial cell. So it becomes folic acid resistant, the sulfur drug resistant. Got my point? Resistant pathways, it develops new resistant pathways so that folic acid can be synthesized, can be obtained from environment. Okay, this is another novel mechanism. Another last mechanism is efflux pump. Here what happens, the bacteria, the plasmid of the bacteria possess some genes The plasmid of the bacteria possess some genes. These genes encode for a protein moiety. These genes encode for a protein moiety. Now what does this protein moiety do? This is actually the efflux pump that I have drawn. And this protein moiety, what it does, suppose antibiotic has already entered, it pushes off the antibiotic out. It pushes the antibiotic out of the cell, extrudes of the antibiotic, or forces the antibiotic out from the cell. Okay. So this is another novel mechanism by which the antibiotic which is inside gets out of the cell. Okay, this is the last mechanism. So you have seen there are several mechanisms by which the bacteria can save itself from the antibiotics. But we guys need to be more smarter than the bacteria so that we can develop a novel drug, so that we can defeat the bacteria and come out with something really good. Okay, I hope you have got my lecture session. You have gained something from this lecture session. Please encourage me by subscribing this channel, Magical Teaching, and do like my channel. Give thumbs up if you really like it, and thank you for watching till the end. Thank you so much.